Welcome to the Deep Dive, your express lane to understanding complex topics. Today, we're tackling reproduction. It's one of the most fundamental processes in life, essential for the survival of a species. Maybe not for an individual survival, but definitely for the species. And it creates just incredible biological variation. We're diving deep into Chapter 17, Reproduction, from Vander's Human Physiology. It's a really core text.
Absolutely. And our mission today is, well, to unpack the key concepts, the mechanisms, the definitions, everything from this chapter. We'll lay it out clearly, step by step, and try to simplify the technical jargon so you can really grasp the fundamentals quickly. Think of it as, your shortcut to being well-informed on this vital topic, those aha moments, without wading through everything yourself.
Okay, so let's unpack this with the basics first. The primary reproductive organs, the gonads, right? Testes in males, ovaries in females. But they're not just there. They have jobs to do.
Exactly. Dual functions, really important. First, they produce the gametes, the reproductive cells, so spermatozoa or sperm, in males, and ova, eggs, in females. And second, they act as endocrine glands. They secrete steroid hormones, the sex hormones.
Like testosterone.
Testosterone is a big one, yes, and androgen. But also estrogens like estradiol and progesterone. And interestingly, both sexes have all of these hormones, just in different predominant amounts.
Got it. Okay, so how are those gametes actually made? Gametogenesis.
Right. It all starts with primordial germ cells. And these cells, they multiply, they proliferate through mitosis.
Mitosis. And that's just cell copying, right? Making identical cells.
Precisely. A cell with 46 chromosomes makes two identical daughter cells, each also with 46. It's about building up the numbers initially. But here's a key difference: the timing. In males, this mitotic activity really ramps up at puberty and, well, basically continues throughout life. In females, it's almost entirely done during fetal development. That's when all the primary oocytes, the precursor egg cells, are produced. No new ones after birth.
Wow, okay, so all the eggs a female will ever have are there from the start.
Yeah.
And then comes meiosis.
Yeah.
This is where it gets really interesting, genetically speaking. Having the chromosomes.
Exactly. Gametes need only 23 chromosomes, not the full 46. Meiosis achieves this through 2 rounds of division. Before the first division, the DNA replicates. So each of the 46 chromosomes now has 2 identical arms, called sister chromatids. Then, the homologous chromosomes, remember, one set came from the mother, one from the father, they pair up. They form these structures called bivalents, and this is crucial. Crossing over happens here. Segments of genetic material are swapped between the non-sister chromatids of these paired chromosomes.
So that shuffles the genetic deck even before the cells divide.
Precisely. It's a major source of genetic recombination, creating unique combinations of genes on each chromosome. Huge for diversity.
Okay, so after crossing over, what happens in the first division?
In the first meiotic division, the homologous chromosomes separate. Not the sister chromatids yet, the pair separate. In males, this yields 2 secondary spermatocytes. In females, it's uneven. 1 large secondary oocyte and a tiny non-functional first polar body. And how these pairs line up and separate is random. That random orientation alone gives you over 8 million possible genetic combinations in the resulting gametes.
Over 8 million, just from that one step.
Yep.
Incredible.
Then comes the second meiotic division. Here, the sister chromatids finally separate. In males, this produces spermatids, which then mature into sperm. In females, it's a bit different. This second division only finishes if fertilization occurs. If it does, you get the mature ovum and another non-functional second polar body. The fertilized egg, the zygote, then has the full 46 chromosomes, 23 from the egg, 23 from the sperm.
So gametogenesis is this incredibly controlled sequence. Division, shuffling, all setting the stage for unique individuals. Hashtag tag 17.2 sex determination.
Yeah.
Okay, we've made the gametes. Now, how is sex actually determined? Who becomes male or female?
That's sex determination, and it's genetic, established right at fertilization. It comes down to the sex chromosomes. Females are XX, males are XY.
So the Y chromosome is the key.
Essentially, yes.
Yeah.
The presence or absence of the Y chromosome. The ovum always brings an X. The sperm brings either an X or a Y. If a Y-bearing sperm fertilizes the egg, it's XY, male. If an X-bearing sperm does, it's XX, female. And interestingly, the sex ratio at birth isn't exactly 50-50. There's a slight consistent edge for males.
Right.
Any idea why? The textbook doesn't delve deep into why, just notes the observation. Oh, and a quick point. In female cells, XX, one X chromosome becomes condensed and largely inactive. It's called a bar body. You can actually see it sometimes under a microscope. We can also visualize all the chromosomes using a technique called karyotyping. Useful for spotting genetic abnormalities like extra or missing sex chromosomes, XXX, XXY, or XO.
Fascinating. So that fundamental XX or XY identity is locked in from the moment of conception. Hashtag 17.3 sex differentiation. Once the genetic blueprint, XX or XY, is set, the body actually has to build the corresponding reproductive system. That's sex differentiation, right?
Exactly. The development of the reproductive structures in the fetus. It starts with the gonads differentiating from a structure called the urogenital ridge. And here's where that Y chromosome really acts. The SRY gene on the Y codes for the SRY protein. This protein is the signal, around the 7th week of gestation, that tells the indifferent gonad to become a testis.
And if there's no SRY gene, no Y chromosome.
Then, in the absence of that SRY signal, the gonad develops into an ovary instead. That's the default pathway, in a sense.
Okay, so that covers the gonads. What about the internal ducts and external bits?
Right. Before the gonads are fully functional, the embryo has both sets of precursor ducts, the Wolffian ducts and the Mullerian ducts, a double system. Now, in genetic males, the newly formed fetal test starts secreting two key things. First, testosterone. This hormone stimulates the Wolffian ducts to develop into the male internal structures epididymis, vas deferens, seminal vesicles. Second, they secrete anti-Mullerian hormone, or AMH. As the name suggests, AMH causes the Mullerian ducts to degenerate, so the female internal structures don't form.
And the external genitalia, penis and scrotum.
That's driven by dihydrotestosterone, or DHT. It's actually derived from testosterone by an enzyme in certain target tissues. DHT is crucial for forming the penis and scrotum. And the testes need to descend into the scrotum, usually late in fetal development. Why? For optimal sperm production later, they need to be about 2 degrees Celsius cooler than core body temperature.
What if they don't descend?
That's a condition called cryptorchidism. It needs to be corrected, usually surgically, to allow for fertility and reduce cancer risk.
Okay, so that's the male pathway. SRY, testes, testosterone plus AMH, male structures. What about females?
In genetic females, XX, there's no SRY gene. So no testes develop. Without testes, there's no testosterone and no AMH produced by the gonads at this critical time. So without testosterone, the Wolffian ducts degenerate. And without AMH, the Mullerian ducts persist and develop into the female internal structures, the fallopian tubes, the uterus, and the upper part of the vagina. The external genitalia also develop along female lines in the absence of DHT. There are also specific ovarian-determining genes on the X chromosome involved in ovary development itself.
It's such a precise cascade. What happens if something goes wrong with these hormonal signals?
Good question. It leads to some well-known conditions. Take androgen insensitivity syndrome. Genetically, these individuals are XY, male, but their cells lack functional receptors for androgens like testosterone and DHT. So, even though the testes make testosterone, the Wolffian ducts don't respond and they regress. AMH is still made, so the Mullerian ducts also regress. And without DHT action, the external genitalia develop as female, so they have a female phenotype despite being XY.
Wow. And are there other examples?
Another is congenital adrenal hyperplasia, or CAH. This commonly involves a genetic defect in cortisol synthesis by the adrenal glands. The body tries to compensate by overproducing precursors, which get shunted into making androgens instead. So if this happens in an XX fetus, they get exposed to abnormally high levels of androgens. This doesn't change the internal organs, ovaries, uteruses, but it can cause virilization of the external genitalia, making them appear ambiguous or more masculine at birth.
These conditions really underscore how critical those specific hormonal signals are at specific times. And thinking bigger picture, this relates to ideas like fetal programming or epigenetics, doesn't it? How early life environment affects later health.
Absolutely. The chapter touches on this. Things like maternal malnutrition or stress during pregnancy can alter the expression of fetal genes, not the genes themselves, but how active they are. This can influence the risk of developing things like high blood pressure or type 2 diabetes much later in life. And these epigenetic changes can sometimes even be passed down to the next generation. It's a really hot area of research.
Yeah, fascinating. And does this hormonal influence extend to the brain?
Yes. The text mentions sexual differentiation of the brain. Hormones present during fetal and early neonatal life can organize brain structures differently between males and females, leading to what are called sexual dimorphisms in the brain, which might underlie some behavioral differences later on. Hashtag 17.4 general principles of reproductive endocrinology.
Okay, we've seen the hormones are crucial. Let's talk about how they're made and controlled, the endocrinology.
Right. The gonadal steroid hormones, androgens like testosterone, estrogens like estradiol and progesterone, they're all synthesized from cholesterol. It's a step-by-step process with specific enzymes at each conversion point determining which hormone is ultimately produced.
Like building blocks.
Exactly. And these hormones typically work by binding to intracellular receptors inside target cells. This hormone receptor complex then binds to DNA and alters the rate of gene transcription, basically turning specific genes on or off more strongly. This changes the synthesis of proteins in the cell, which causes the hormone's effects.
So who's the master controller of all this, pulling the strings?
That would be the hypothalamo-pituitary gonadal axis, often called the HPG axis. It's a classic endocrine feedback loop.
Okay, break that down for us. Hypothalamus.
The hypothalamus, a part of the brain, releases gonadotropin-releasing hormone, or GnRH.
GnRH, gonadotropin releasing, so it releases gonadotropins.
Precisely. GnRH travels a short distance to the anterior pituitary gland and stimulates it to release two gonadotropins, follicle-stimulating hormone, FSH, and luteinizing hormone, LH. FSH and LH then travel through the bloodstream to the gonads, testes, or ovaries.
And what do FSH and LH do with the gonads?
They stimulate gonadal function. Broadly, they promote gamete maturation, spermatogenesis or follicle development, and stimulate the secretion of the sex hormones, like testosterone or estrogen and progesterone. A really important point is that GnRH from the hypothalamus is released in pulses, not continuously. This pulsatile pattern is essential for the pituitary to respond properly. Constant high GnRH can actually shut down FSH and LH release.
Interesting, like it needs breaks. And what about feedback? Do the gonadal hormones talk back to the brain?
Absolutely. That's crucial for regulation. The gonadal steroids, testosterone, estrogen, progesterone, generally exert negative feedback. They inhibit the secretion of GnRH from the hypothalamus and also reduce the sensitivity of the pituitary to GnRH, thus lowering FSH and LH release. There's another player too, inhibin. It's a hormone secreted by the gonads, specifically Sertoli cells in males, granulosa cells in females, that specifically inhibits the secretion of FSH from the pituitary but not LH.
So multiple layers of control. But
Yeah.
you mentioned something earlier. Estrogen sometimes does the opposite.
Yeah.
Positive feedback.
Yes, that's a key feature, particularly in the female cycle. At a very specific point, when estrogen levels get very high for a sustained period, it flips the switch and actually stimulates GnRH release and makes the pituitary more sensitive to GnRH. This causes a surge in LH and FSH. We'll see how critical this is later.
Wow, okay, that's a twist.
And just keep in mind, overall reproductive hormone levels aren't constant throughout life. They're actually quite high during fetal development, especially in males, then very low during childhood. Then they surge dramatically at puberty, driving sexual maturation. In women, they cycle monthly. And finally, they decline with aging.
All right, let's zoom in on the male system now. We got the two tests, obviously, but then this whole network of ducts and glands, plus the penis. Let's start with the testes location in the scrotum. Why outside the main body cavity?
It's all about temperature. As we mentioned, spermatogenesis, the process of making sperm, is very temperature sensitive. It works optimally at about 2 degrees Celsius below core body temperature. The scrotum provides this cooler environment.
It's clever design. Okay, inside the testes.
Inside, they're packed with tiny coiled tubes called the seminiferous tubules. This is ground zero for sperm production. Miles of them if you stretch them out. From the seminiferous tubules, sperm move into a network called the ret testes, then through efferent ductules into the epididymis.
The epididymis, that's for storage and maturation.
Exactly. Sperm spend time there, maturing and gaining motility. From the epididymis, they enter the vas deferens, or ductus deferens. This tube travels up, loops around the bladder, and then joins with the duct from the seminal vesicle to form the ejaculatory duct. The two ejaculatory ducts then pass through the prostate gland, which sits just below the bladder and surrounds the urethra.
So the ejaculatory ducts empty into the urethra within the prostate.
Correct. And just below the prostate, the bulbourethral glands, or Cowper's glands, also add their secretions to the urethra. The urethra then runs through the penis.
So sperm travel quite a distance. All those glands, seminal vesicles, prostate, bulbourethral, they add fluids.
Yes. Their secretions combine with the sperm to form semen.
And what's in those fluids? Just
Yeah.
volume.
Much more than volume. The seminal vesicles provide fructose for energy, prostaglandins, which might help sperm motility and uterine contractions, and substances that help semen clot temporarily after ejaculation. The prostate adds citrate, another nutrient, enzymes like prostate-specific antigen, PSA, that help liquefy the semen later, and zinc. And the bulbourethral glands secrete a clear alkaline mucus just before ejaculation, which helps lubricate the urethra and neutralize any acidic urine residue. It's a complex cocktail designed to nourish, protect, and transport the sperm. Hashtag tag tag 17.6 spermatogenesis.
Okay, let's go back inside those seminiferous tubules. How does a basic stem cell turn into a highly specialized sperm cell? That whole spermatogenesis journey.
Right. It starts with the stem cells called spermatogonia. They line the outer edge of the tubule. Some undergo mitosis to constantly replenish the stem cell pool. Others commit to becoming sperm. A committed spermatogonium differentiates into a primary spermatocyte. This cell still has 46 chromosomes, and it's ready for meiosis. The primary spermatocyte undergoes the first meiotic division, meiosis the first, to produce two secondary spermatocytes, each now with 23 chromosomes, but each chromosome still has two chromatids.
Then meiosis the second?
Then meiosis the second happens quickly. Each secondary spermatocyte divides into two spermatids. So one primary spermatocyte ultimately yields four spermatids, each with 23 single chromatid chromosomes.
But spermatids aren't sperm yet, are they? They look different.
Correct. The final stage is spermiogenesis. This isn't cell division, it's a dramatic physical transformation, a differentiation process. The round spermatid remodels itself into the characteristic shape of a spermatozoon, or sperm. You get the head, containing the nucleus with the 23 chromosomes, capped by the acrosome, a vesicle packed with enzymes needed to penetrate the egg. Then the midpiece, which is loaded with mitochondria to generate ATP for movement. And finally, the long tail, or flagellum, that propels the sperm.
That whole process, spermatogonium to spermatozoon, how long does that take?
It takes roughly 64 days, and it's happening continuously in different sections of the tubules, resulting in the production of about 30 million sperm per day in a healthy young adult male. It's a massive output.
Wow. Who's managing this whole factory floor? Are there support cells?
Absolutely essential are the Sertoli cells. These are large cells that extend from the base to the lumen of the seminiferous tubule, surrounding the developing germ cells at every stage.
What do they do?
Lots of things. They form tight junctions between themselves, creating the blood tests barrier. This isolates the developing sperm, which are genetically different from the body, from the immune system, and harmful substances in the blood. They nourish the developing germ cells, basically feeding them. They secrete the fluid that fills the tubule lumen and helps transport sperm out. They also secrete androgen-binding protein, ABP, into the lumen. ABP binds testosterone, keeping local levels incredibly high right where spermatogenesis needs it. And importantly, Sertoli cells respond to hormones, specifically FSH from the pituitary and testosterone from neighboring Leydig cells to regulate the rate of spermatogenesis.
So they're like the nurse cells and managers combined.
That's a good way to think of it. They also produce inhibin, the hormone we mentioned earlier that provides negative feedback, specifically on FSH secretion.
Okay, you mentioned Leydig cells providing testosterone. Where are they?
The Leydig cells, also called interstitial cells, are located in the connective tissue between the seminiferous tubules. Their main job is to synthesize and secrete testosterone in response to LH from the pituitary.
Ah, so the sperm production happens inside the tubules with Sertoli cell help, and the testosterone production happens outside the tubules by Leydig cells. A nice separation of functions.
Exactly. Though the testosterone produced by Leydig cells is crucial for the Sertoli cells inside the tubules to support spermatogenesis. Hashtag tag 17.7, transport of sperm, erection, and ejaculation.
Okay, sperm are made. Now they need to get out and, well, potentially fulfill their function. How do they move initially?
From the seminiferous tubules, they're flushed along by the fluid secreted by the Sertoli cells, moving into the rete testes and then the epididymis. They're not really swimming on their own yet. In the epididymis and the vas deferens, movement is mainly due to peristalsis, rhythmic, smooth muscle contractions in the walls of the ducts. By the time they reach the end of the vas deferens, they're mature, concentrated, and stored, ready for ejaculation.
And this is where a vasectomy intervenes, right?
Yes. A vasectomy involves cutting or blocking the vas efferens, usually in the scrotum. It simply prevents sperm from being transported out during ejaculation. It doesn't affect testosterone production by the Leydig cells or sperm production in the testes initially.
Okay, makes sense. Now the events leading up to ejaculation, erection. How does that work? It seems like a plumbing issue, but it's more complex.
It's definitely neurovascular. The penis contains cylinders of erectile tissue with lots of vascular spaces, like sponges. Erection happens when the small arteries supplying these spaces dilate, allowing much more blood to flow in than flows out through the compressed veins. This engorgement causes the penis to become rigid.
What triggers the arteries to dilate? Nerves?
Yes, primarily nerves. It involves inhibition of the normally active sympathetic vasoconstrictor nerves and activation of parasympathetic nerves. Crucially, these parasympathetic nerves and perhaps other non-adrenergic, non-cholinergic autonomic neurons release nitric oxide, or NO.
Nitric oxide, like the gas.
Exactly. NO acts locally on the smooth muscle cells in the arterial walls. It activates an enzyme that produces cyclic GMP, or cGMP. And cGMP is the messenger that causes the smooth muscle to relax, leading to vasodilation and increased blood flow. Higher brain centers, of course, play a huge role in initiating or inhibiting erections via these descending pathways.
So if that NOC-GMP pathway doesn't work well, that leads to problems.
Precisely. That's the basis for erectile dysfunction, or ED. And it's why drugs like sildenafil Viagra work. They are PDE5 inhibitors.
PDE5, phosphatidylserine type 5, what does that do?
PDE5 is the enzyme that normally breaks down cPMP. So by inhibiting PDE5, these drugs allow cGMP levels to stay higher for longer when NO is released during sexual stimulation. This prolongs the smooth muscle relaxation and enhances the ability to achieve and maintain an erection. They don't cause an erection without stimulation, but they enhance the natural process.
Got it. Okay, so after erection comes ejaculation.
Ejaculation is primarily a spinal cord reflex, triggered by sufficient stimulation. It happens in two phases. First is emission. This is controlled by the sympathetic nervous system. Smooth muscles in the epididymis, vas deferens, seminal vesicles, and prostate gland contract, emptying sperm and glandular secretions into the urethra. At this point, the semen is in the urethra at the base of the penis. The second phase is expulsion. This involves rapid, rhythmic contractions of the smooth muscle in the urethra and skeletal muscles at the base of the penis. This forcefully expels the semen out.
And the body makes sure urine doesn't mix in.
Yes, during emission and ejaculation, the sphincter muscle at the base of the bladder contracts tightly, preventing urine from entering the urethra and also preventing semen from going backward into the bladder.
And the whole experience, the intense pleasure and physiological changes, that's orgasm.
Yes, orgasm typically accompanies ejaculation in males, involving widespread physiological changes and intense sensation. Hashtag 17.8 hormonal control of male reproductive functions.
We touched on the HPG axis earlier. Let's tie it specifically to male functions now. Remind us of the chain of command.
Okay. Hypothalamus releases GnRH in pulses. Anterior pituitary responds by releasing LH and FSH. LH primarily travels to the Leydig cells in the testes and stimulates them to produce and secrete testosterone. FSH primarily targets the Sertoli cells inside the seminiferous tubules. It acts along with testosterone to stimulate spermatogenesis and also prompts Sertoli cells to release inhibin and ABP.
So LH Leydig cells, nodal testosterone. FSH Sertoli cells, nodal spermatogenesis support, plus inhibin, simple enough. And the feedback loops. How does the system regulate itself?
Testosterone is the main feedback signal. It acts back on the hypothalamus to decrease the amplitude of GnRH pulses, and it acts on the anterior pituitary to make it less responsive to GnRH, specifically reducing LH secretion. So high testosterone dampens its own production signal.
Negative feedback. What about FSH?
That's where inhibin comes in. Inhibin, secreted by the Sertoli cells in response to FSH, travels to the anterior pituitary and specifically inhibits FSH secretion without significantly affecting LH.
So testosterone mainly controls LH, and inhibin mainly controls FSH, a dual control system.
That's a good way to summarize it. It allows finer tuning of sperm production versus testosterone levels.
Okay, let's talk more about testosterone itself. It does more than just regulate its own production and help sperm develop, right?
Oh, absolutely. Testosterone has wide-ranging effects throughout the body. The textbook has a great table, Table 17.3, summarizing them. Besides being essential locally within the testes for spermatogenesis, it's responsible for inducing and maintaining the male accessory reproductive organs, seminal vesicles, prostate, etc. It drives the development of male secondary sex characteristics at puberty, deepening voice, beard growth, male pattern hair growth, and ironically, later baldness in susceptible men. It has significant anabolic effects, stimulating protein synthesis, especially in skeletal muscle, leading to increased muscle mass. It also promotes bone growth during puberty, but eventually causes the epiphyseal plates to close, stopping height growth.
And behavior.
Yes, it's important for sex drive or libido in males. And it also stimulates the secretion of erythropoietin, the hormone that boosts red blood cell production.
That's a lot of roles. You mentioned earlier that testosterone can be converted into other hormones.
Right. In certain target tissues, testosterone isn't the final active hormone. An enzyme called 5-alpha reductase converts testosterone into the more potent androgen dihydrotestosterone, DHT. DHT is particularly important for the external genitalia development, prostate growth, and male pattern hair growth loss. In other tissues, notably the brain and adipose tissue, another enzyme called aromatase converts testosterone into estradiol and estrogen.
Estrogen in men from testosterone.
Yes. And this locally produced estrogen seems to be important for certain brain functions, bone health, and feedback regulation. So testosterone can act directly or via conversion to DHT or via conversion to estradiol, depending on the target cell and the enzymes it contains.
That explains why treatments targeting those enzymes exist, like 5-alpha reductase inhibitors for prostate enlargement or baldness.
Exactly. Or aromatase inhibitors used in some contexts. It highlights the complexity of androgen action. Hashtag 17.9 puberty, male.
Right, let's talk about puberty. That period of dramatic change when reproductive function awakens. When does it typically happen in boys?
Usually between the ages of 12 and 16, some of the very first signs, like maybe some pubic or axillary hair, might actually be due to weak androgens produced by the adrenal glands slightly earlier. But the main event of puberty is driven by the reactivation and amplification of the HPG axis. The brain essentially becomes less sensitive to the negative feedback effects of testosterone.
So the brakes come off a bit.
In a way, yes. This allows GnRH pulse frequency and amplitude to increase significantly. That drives up LH and FSH, which in turn stimulates the testes to produce much more testosterone. And it's the surge in testosterone and its conversion product DHT that drives most of the changes we associate with male puberty.
The secondary sex characteristics we mentioned.
Yes. Deepening voice due to larynx growth, growth of facial and body hair, increased skin oil production leading to acne, development of the penis and scrotum, achieving reproductive capability, first ejaculation. Also, the characteristic male pattern of fat distribution, the pubertal growth spurt in height, driven by testosterone and growth hormone, and the increase in muscle mass testosterone is a potent anabolic steroid in this regard. It also increases erythropoietin, leading to higher hematocrit in males than females.
And sex drive.
Androgens are definitely essential for developing and maintaining male sex drive or libido during and after puberty.
You mentioned anabolic steroids. What about abuse?
Yes, the text briefly notes the serious health risks associated with abusing synthetic anabolic steroids. Besides liver damage, mood changes, and cardiovascular issues, they exert powerful negative feedback on the HPG axis. This shuts down natural GnRH, LH, and FSH production, leading to decreased endogenous testosterone secretion, testicular atrophy, shrinking, and infertility. The body basically thinks it has way too much testosterone already. Hashtag 17.B1 hypogonadism.
What if the system doesn't function properly? Subnormal testicular function. Hypogonadism.
Right. Hypogonadism just means decreased function of the gonads, either in terms of spermatogenesis or testosterone production, or both. It can be classified based on where the problem lies. Primary hypogonadism means the problem is in the tests themselves. They're failing to respond properly to LH and FSH. Secondary hypogonadism means the testes are potentially fine, but they're not getting the right stimulation from the pituitary, so LH and FSH levels are low.
Can you give examples?
Sure. A classic example of primary hypogonadism is Klinefelter syndrome. These individuals have an extra X chromosome, so their genotype is 47,000 XXY. It's usually caused by an error during meiosis called non-disjunction. They tend to have small, firm tests that don't produce much testosterone or sperm. This leads to incomplete masculinization, often with features like reduced body hair, some breast enlargement, gynecomastia, and infertility. Because the testes aren't producing testosterone, the negative feedback is lost, so LH and FSH levels are actually high.
Okay, and secondary hypogonadism.
This could be caused by anything that damages the pituitary or hypothalamus, reducing LH and FSH secretion, for example, pituitary tumors. Or, specifically, hyperprolactinemia, abnormally high levels of the hormone prolactin, often from a pituitary tumor, can suppress GnRH release and thus LH and FSH. General hypopituitarism, decreased function of the entire anterior pituitary, would also cause it. Even things like opiate abuse can suppress the HPG axis. Treatment for hypogonadism often involves hormone replacement therapy, usually testosterone. Hashtag 17.11 andropause.
As men get older, does function just stop, like menopause in women, or is it different?
It's generally more gradual. The term's sometimes used as andropause, or the male climacteric. Starting around age 40, there's a slow but steady decline in testosterone secretion. This seems to reflect both some deterioration within the tests themselves and possibly decreased responsiveness to LH and FSH over time.
What are the effects?
It can lead to symptoms like decreased libido, reduced muscle mass and strength, increased fat mass, sometimes changes in mood or cognition, like depression. Sperm production might decrease and sperm motility might lessen, but many men remain fertile into old age. It's quite variable compared to the more definitive cessation of cycles in female menopause.
Right. Let's switch gears completely now and dive into the female reproductive system. You mentioned earlier it operates on cycles.
Yes, that's a fundamental difference. Instead of continuous gamete production, like in males, female reproduction is characterized by cyclical changes. The maturation and release of an egg, ovulation, is timed within the menstrual cycle. The average cycle length is about 28 days, and by convention, day 1 is the first day of menstrual bleeding.
Okay, so what are the main parts of this system? The internal genitalia.
You have the 2 ovaries, the primary gonads, then the 2 fallopian tubes, or oviducts, which extend from near the ovaries towards the uterus. The open ends near the ovaries have finger-like projections called fimbriae that help capture the ovulated egg. The fallopian tubes lead into the uterus, a muscular organ where a fetus develops. The lower, narrow part of the uterus is the cervix, which opens into the vagina, the canal that connects the uterus to the outside. An important anatomical note. Unlike in males where the urinary and reproductive tracts merge, in females, the urethra opens separately, anterior to the vagina.
Got it. And the external genitalia?
Collectively called the vulva. This includes the mons pubis, fatty tissue over the pubic bone, the labia majora, outer folds, homologous to the scrotum, the labia minora, inner folds, the clitoris, anteriorly homologous to the penis, composed of erectile tissue, and the vestibule, which is the area enclosed by the labia minora, containing the openings of the urethra and vagina. There are also vestibular glands that provide lubrication, hashtag 17.13 ovarian functions.
So the ovaries are the gonads. What are their main jobs? Similar to the testes.
Broadly, yes. They perform oogenesis, producing the female gametes, the ova, or eggs. They mature the oocyte, they release it at ovulation, and, like the testes, they secrete steroid hormones, primarily estrogen and progesterone, as well as the peptide hormone inhibin. A key structural point. The eggs develop within structures called follicles inside the ovary. After ovulation, the remnants of the follicle transform into a structure called the corpus luteum. These structures are central to the ovarian cycle.
Let's trace oogenesis again, the production of the egg. You said it starts before birth.
Yes, crucially. During fetal development, the primordial germ cells undergo mitosis to produce millions of primary oocytes. These primary oocytes then begin meiosis, but they arrest partway through. They pause in meiotic prophase. A female is born with her lifetime supply of these arrested primary oocytes, maybe 2 to 4 million. No new ones are formed after birth.
So they just sit there for years, decades.
Exactly, and most of them actually degenerate over time through a process called atresia. From the millions present at birth, maybe only 400 or so will ever actually be ovulated during her reproductive years.
That means eggs ovulated later in life are biologically much older. Does that matter?
Yes, it's thought to be linked to the increased incidence of chromosomal abnormalities like Down syndrome in pregnancies of older women. The meiotic machinery might be more prone to errors after decades of arrest.
Okay, so the primary oocyte is arrested. When does it finish meiosis?
Just before ovulation, triggered by hormonal signals, the primary oocyte selected for that cycle completes its first meiotic division. Remember, this division is unequal. It produces one large secondary oocyte, which contains most of the cytoplasm, and one tiny first polar body, which is basically just discarded nuclear material. This secondary oocyte then begins meiosis synthesis, but arrests again, this time in metaphase 2.
And it only finishes meiosis ascendis.
If it gets fertilized by a sperm. Fertilization triggers the completion of meiosis II, again producing one large cell, the mature ovum, and a tiny second polar body. The nucleus of the ovum then fuses with the sperm nucleus to form the zygote.
So unlike males, where one primary spermatocyte yields 4 sperm, here one primary oocyte yields only one functional ovum.
Correct. All the cytoplasm and nutrients are conserved for that single potential offspring.
You mentioned follicles. How do they develop alongside the oocyte?
The oocyte doesn't exist alone. It's always encased in supporting cells. It starts as a primordial follicle, the primary oocyte surrounded by a single layer of flat cells called granulosa cells. As development begins, even before puberty, some start growing, the granulosa cells multiply and become cuboidal. They start secreting estrogen and inhibin. A protective layer called the zona pellucida forms around the oocyte itself, secreted by both the oocyte and granulosa cells. Connective tissue cells outside the granulosa layer differentiate into the perdita cells. The penis cells work together with granulosa cells to produce estrogen. The penis cells make androgens, which diffuse to the granulosa cells where an enzyme, aromatase, converts them to estrogen. As the follicle grows further, fluid begins to accumulate between the granulosa cells, eventually forming a large, fluid-filled cavity called the antrum. Now it's an antral follicle.
Do all follicles reach this stage?
Many follicles start developing in each cycle, reaching the preantral and early antral stages. But usually under the influence of hormones at the beginning of a menstrual cycle, only about 10 to 25 of these begin to enlarge further. And then, through a selection process we'll discuss, typically only one follicle becomes the dominant follicle. It continues to grow rapidly, while the others undergo atresia, they degenerate. This mature, dominant follicle ready for ovulation is sometimes called a Graafian follicle.
And ovulation is the release of the egg from this dominant follicle.
Yes. Around day 14 of a typical cycle, the follicle wall and the ovarian wall overlying it rupture, releasing the secondary oocyte, still surrounded by some granulosa cells, out of the ovary.
What happens to the follicle left behind?
After ovulation, the remaining granulosa and the theca cells of the ruptured follicle are transformed, under the influence of LH, into the corpus luteum, which means yellow body. The corpus luteum becomes a temporary endocrine gland, secreting large amounts of progesterone as well as significant amounts of estrogen and inhibin for about 10 to 14 days.
And if pregnancy doesn't happen?
If there's no pregnancy signal, the corpus luteum degenerates towards the end of the cycle.
So the ovarian cycle has these two main phases then. The follicular phase, from menstruation to ovulation, dominated by the growing follicle and estrogen.
Correct.
And the luteal phase, after ovulation, dominated by the corpus luteum and progesterone.
Exactly. Follicular phase is days 114 roughly. Luteal phase is days 1428 roughly. #TAG 17.14, control of ovarian function.
Okay, this sounds like it needs some serious hormonal coordination. The HPG axis, again, but maybe working differently than in males.
Very differently, especially with that positive feedback element. The whole system relies on GnRH pulses from the hypothalamus, just like in males, but the frequency and amplitude of these pulses change dramatically throughout the menstrual cycle, driving the changes in FSH and LH. Let's walk through the typical hormonal pattern, maybe referencing figure 17.22 in the textbook.
Okay, starting at the beginning of the cycle, day 1, menstruation is happening. What are the hormones doing?
At the very end of the previous cycle, the corpus luteum degenerated, so estrogen and progesterone levels crashed. This removes the negative feedback on the pituitary. As a result, FSH levels start to rise slightly in the early follicular phase. This rise in FSH is the signal that rescues a group of those 10-25 early antral follicles and stimulates them to grow further.
So FSH kicks things off. What does it do with the follicles?
FSH primarily acts on the granulosa cells. It stimulates them to multiply, increases their production of estrogen, especially by boosting aromatase activity, and promotes the formation and enlargement of the antrum. Meanwhile, LH levels are still relatively low, but are essential for stimulating the theca cells to produce androgens, which the granulosa cells then convert to estrogen.
So estrogen levels start to rise as these follicles grow.
Yes. And initially, this rising estrogen exerts negative feedback on the pituitary, primarily suppressing FSH secretion. Inhibin from the granulosa cells also contributes significantly to suppressing FSH. This drop in FSH is thought to be crucial for selecting the dominant follicle. The follicle that becomes most sensitive to FSH, or maybe produces the most estrogen locally, is the one that can continue to thrive even as FSH levels fall. The others, deprived of sufficient FSH stimulation, undergo atresia.
Survival of the fittest follicle. So the dominant follicle keeps growing, pumping out more and more estrogen.
Exactly. And this leads us to the critical turning point, the late follicular phase and the LH surge. As estrogen levels produced by the dominant follicle climb higher and higher and stay high for a day or two, estrogen's effect on the pituitary flips from negative to positive feedback.
The switch.
High sustained estrogen makes the anterior pituitary more sensitive to GnRH, and it might also stimulate the hypothalamus to release more GnRH. The result is a massive, explosive release of LH from the pituitary, the LH surge. There's also a smaller surge in FSH.
And this LH surge is the trigger for?
It triggers a cascade of events within the dominant follicle detailed in Table 17.5. First, it causes the primary oocyte to finally complete meiosis cycle and become a secondary oocyte. It stimulates further swelling of the follicle, increasing antrum size and blood flow. It temporarily halts estrogen synthesis by the granulosa cells and causes them to start producing some progesterone even before ovulation. Crucially, it triggers the release of enzymes and prostaglandins within the follicle that break down the follicle wall and the overlying ovarian tissue, leading to the rupture ovulation about 24-36 hours after the surge begins. And finally, the LH surge promotes the transformation of the remaining granulosa and the deca cells into the corpus luteum.
Wow, so that LH surge is the absolute linchpin of the cycle.
It really is. Everything pivots around it.
Okay, so ovulation happens around day 14. Now we're in the luteal phase. The corpus luteum is formed. What does it do?
The corpus luteum, initially maintained by the lingering effects of the LH surge and then low basal levels of LH, becomes a progesterone-producing powerhouse. It also secretes significant amounts of estrogen and inhibin.
Progesterone is the dominant hormone now.
Yes, and these high levels of progesterone, along with the estrogen, exert strong negative feedback on the hypothalamus and pituitary. This suppresses GnRH, LH, and FSH secretion back down to low levels.
Why suppress FSH and LH now?
This prevents any new follicles from developing during the luteal phase, and it prevents another LH surge. You only want one ovulation per cycle, typically.
Makes sense. So the corpus luteum keeps pumping out hormones, maintaining the second half of the cycle. But what if pregnancy doesn't occur?
If the egg isn't fertilized and doesn't implant, there's no signal, like the pregnancy hormone HCG, which we'll get to maintain the corpus luteum. It has an intrinsic lifespan of about 10-14 days. So towards the end of the cycle, around day 24-26, it starts to degenerate. As the corpus luteum degenerates, its production of progesterone, estrogen, and inhibin plummets.
Ah, and the drop in hormones removes the negative feedback.
Exactly. With the negative feedback lifted, FSH and LH levels start to rise again. And that rising FSH initiates the growth of the next cohort of follicles, starting the whole cycle over again on day 1 with menstruation.
It's such an elegant loop. The ovarian events drive the hormones, which drive the next ovarian events. Incredible orchestration. Hashtag TASH, hashtag 17.15 uterine changes in the menstrual cycle. Okay, while all this drama is happening in the ovary, the uterus isn't just sitting idle. It's changing too, right? In preparation for a potential pregnancy.
Absolutely. The changes in the uterine lining, the endometrium, are directly controlled by the fluctuating levels of estrogen and progesterone produced by the ovaries. We typically divide the uterine cycle into 3 phases that correspond to the ovarian phases.
Let's start with day 1. Ovarian follicular phase is just beginning. Corpus luteum from the last cycle is gone. What's the uterus doing?
This is the menstrual phase, lasting roughly from day 1 to day 5. Because estrogen and progesterone levels are very low due to the corpus luteum's demise, the endometrial lining, which had built up in the previous cycle, loses its hormonal support. Blood vessels in the lining constrict, the tissue dies, and it sluices off, resulting in the menstrual bleeding. This bleeding marks the start of the new cycle.
Okay, so menstruation is the shedding of the old lining. What happens next as the follicles start growing and making estrogen?
As estrogen levels rise during the mid to late follicular phase, roughly day 5 to day 14, we enter the proliferative phase of the uterine cycle. Estrogen stimulates the regrowth of the endometrium. Cells multiply, the lining thickens again, glands reform, and new blood vessels grow. Estrogen also stimulates the growth of the underlying uterine smooth muscle, the myometrium. Another effect of rising estrogen in this phase is on the cervical mucus. It becomes abundant, clear, and watery, which is thought to facilitate the passage of sperm through the cervix into the uterus around the time of ovulation.
So the uterus is rebuilding, preparing. Then ovulation happens around day 14. What happens in the uterus during the ovarian luteal phase?
After ovulation, the corpus luteum forms and starts pumping out progesterone, along with estrogen. This shifts the uterus into the secretory phase, lasting from ovulation to the start of the next menstruation, roughly day 14 to day 28. Progesterone is the key player here. It acts on the estrogen-primed endometrium, transforming it into a highly vascularized, glandular, nutrient-rich tissue. The glands start secreting glycogen, enzymes, and other substances, essentially creating a welcoming, nourishing environment suitable for the implantation of a fertilized egg. Progesterone also has other important effects. It inhibits contractions of the myometrium, keeping the uterus quiet, which is important for maintaining a potential pregnancy. And it changes the cervical mucus again, making it thick, sticky, and scanty. This forms a mucus plug in the cervix, which helps block bacteria and sperm from entering the uterus during the luteal phase in pregnancy.
So the secretory phase is all about making the uterus receptive and maintaining potential pregnancy.
Exactly, preparing the perfect nest.
And if implantation doesn't occur, the corpus luteum dies, progesterone and estrogen fall, and that triggers menstruation again.
Precisely. The withdrawal of progesterone and estrogen support leads to the events causing menstruation. The constriction of uterine blood vessels, mediated partly by prostaglandins produced locally in the endometrium, leads to oxygen deprivation, tissue death, and shedding. Those prostaglandins also stimulate contractions of the myometrium, which helps expel the menstrual debris but can also cause the cramps associated with menstruation dysmenorrhea. Hashtag 17.16 additional effects of gonadal steroids.
We focused on the reproductive cycle, but estrogen and progesterone must have effects elsewhere in the body too, like testosterone in males.
Oh, definitely. Table 17.8 in the book gives a good overview. Estrogen has widespread effects. During puberty, it stimulates the growth of the reproductive tract, uterus, fallopian tubes, vagina, and the external genitalia. It causes breast development, specifically promoting duct growth and fat deposition. It contributes to the typical female body shape and fat distribution. It generally has beneficial effects on skin, anti-acne compared to androgens. It promotes bone growth during puberty, but like testosterone, eventually leads to the closure of the epithelial plates. Crucially, it helps maintain bone density throughout reproductive life, protecting against osteoporosis.
Osteoporosis, loss of bone density, that becomes a bigger issue after menopause when estrogen drops.
Exactly. Estrogen also has vascular effects. Its withdrawal at menopause is linked to the hot flashes many women experience. It stimulates the pituitary to secrete prolactin, but interestingly, it inhibits prolactin's action on the breast in terms of milk production during pregnancy. And there's evidence it has protective effects against atherosclerosis before menopause.
Wow, quite a range. What about progesterone, besides its uterine effects?
Progesterone's effects are generally more focused on reproduction and pregnancy. It does stimulate the growth of the glandular tissue in the breast, complementing estrogen's effect on ducts. Like estrogen, it inhibits prolactin's milk-producing action during pregnancy. A well-known systemic effect is that it increases body temperature slightly, by about 0.5 degrees Celsius after ovulation. This temperature shift can be used as an indicator that ovulation has occurred. In some tissues, progesterone can also oppose estrogen's effects. It's sometimes described as having an anti-estrogen action, for example, decreasing the proliferation of vaginal epithelial cells that estrogen stimulates.
This complexity makes you wonder about things like premenstrual syndrome, PMS, or the more severe PMDD, premenstrual dysphoric disorder. Are they just hormone imbalances?
It's complex. Women experiencing these symptoms, things like anxiety, irritability, depression, bloating, breast tenderness appearing in the luteal phase and resolving with menstruation, usually have normal plasma concentrations of estrogen and progesterone. So it's likely not a simple excess or deficiency, but maybe an abnormal response of the central nervous system to the normal cyclical fluctuations of these hormones, possibly involving interactions with neurotransmitters like serotonin. The exact causes are still being researched.
And what about androgens in women? Do women have testosterone?
Yes, they do. Small amounts are produced by the ovaries and also by the adrenal glands. These androgens are important for stimulating the growth of pubic and axillary hair at puberty and are thought to be the primary driver of sex drive, libido, in women. If androgen levels become excessively high in women, for example, due to certain ovarian or adrenal tumors or conditions like polycystic ovary syndrome, it can lead to virilization development of male-like characteristics such as facial hair, deepening voice, and muscle bulk. Hashtag 17.17 puberty, female.
We talked about male puberty. What about female puberty? Does it start earlier?
Yes, typically it begins earlier than in males, usually between the ages of 8 and 13. Similar to males, the trigger seems to be a change in brain function that leads to increased pulsatile secretion of GnRH from the hypothalamus. The exact mechanisms aren't fully understood, but neurons releasing a peptide called kisspeptin seem to play a crucial role in stimulating GnRH neurons. There's also evidence linking the onset of puberty to metabolic signals, specifically the hormone leptin, which is released by adipose tissue.
Leptin, from fat cells, so body fat influences puberty timing.
It appears so. Sufficient body fat stores seem necessary for puberty to begin and for menstrual cycles to be maintained. This might explain why girls with very low body fat due to excessive exercise or conditions like anorexia nervosa often experience delayed puberty or amenorrhea, the absence of menstrual cycles.
Interesting link. So increased GnRH drives up FSH and LH, which stimulates the ovaries.
Right. The ovaries start producing significant amounts of estrogen. And it's this estrogen that drives the development of the female secondary sex characteristics. Breast development, growth of the internal and external genitalia, the pubertal growth spurt, though typically less dramatic than in males, and the characteristic female pattern of fat deposition, hips, thighs.
When does the first period menarche happen in all this?
Menarche is actually a relatively late event in puberty, occurring on average around 12.5 years of age in the U.S. It indicates that the hormonal cycles are becoming established, but the initial cycles are often irregular and anovulatory without ovulation. Regular ovulation typically starts somewhat later.
What if puberty happens too early?
That's called precocious puberty, the appearance of secondary sex characteristics before age 8 in girls, or 9 in boys. It can have various causes, sometimes related to early activation of the HPG axis or, less commonly, hormone-secreting tumors. While these children might be tall initially, the early exposure to sex steroids often causes the epiphyseal plates in their bones to close prematurely, resulting in a shorter final adult height. Hashtag 17.18 female sexual response.
Similar to males, there's a physiological sexual response in females.
Yes. During sexual excitement, there's vasodilation and engorgement of erectile tissues. This includes the clitoris becoming erect and increased blood flow to the breasts, causing nipple erection, and the vaginal walls. This increased blood flow to the vagina also leads to the secretion of lubricating fluid from the vaginal epithelium.
And orgasm.
Orgasm in females is characterized by intense, pleasurable sensations and rhythmic contractions of pelvic muscles, including the uterus and vaginal walls, along with systemic changes like increased heart rate and blood pressure. Unlike males, females do not have a refractory period immediately after orgasm and can potentially experience multiple orgasms.
You mentioned earlier that libido in women might be more related to androgens.
Yes, while estrogen is crucial for the physical changes in lubrication, sexual desire or libido in women seems to be more closely linked to androgen levels from the ovaries and adrenals than to estrogen levels. Hashtag 17.19 menopause.
Eventually, reproductive cycles stop. This transition is menopause, right?
Yes, it marks the end of a woman's reproductive capacity. The period leading up to it, characterized by increasingly irregular cycles, is called perimenopause, which can last several years. Menopause itself is formally defined as the cessation of menstruation for at least 12 consecutive months. The average age is around 51, 52, but it varies.
What's the underlying cause? Why do the ovaries stop working?
The primary cause is essentially ovarian failure. The ovaries run out of viable follicles. Remember, a woman is born with all her potential eggs, and they diminish over time through ovulation and atresia. As the number of follicles dwindles, the ovaries become less responsive to stimulation by FSH and LH. They produce less and less estrogen and inhibin.
So estrogen levels drop significantly.
Markedly. And because estrogen and inhibin levels fall, the negative feedback on the pituitary is greatly reduced. As a result, the pituitary tries to whip the failing ovaries into action by pumping out more FSH and LH.
So paradoxically, menopause is characterized by very low estrogen, but very high levels of FSH and LH.
And the low estrogen causes the symptoms associated with menopause. Yes. The lack of estrogen leads to atrophy, thinning and drying, of the vaginal lining and urethral tissues, which can cause discomfort and urinary issues. Breasts may decrease in size. The loss of estrogen's protective effect on bone leads to accelerated bone density loss and increased risk of osteoporosis. Hot flashes, sudden sensations of intense heat, often with sweating and flushing, are very common and are related to estrogen withdrawal affecting thermoregulation centers in the brain. Sleep disturbances are also frequent. Furthermore, the risk of cardiovascular disease increases significantly after menopause, partly due to the loss of estrogen's protective vascular effects.
This is why hormone replacement therapy, HRT, was, and sometimes still is, used to replace the lost estrogen.
Exactly. Estrogen therapy, often combined with progesterone if the woman still has her uterus to protect the endometrium, can be very effective at relieving symptoms like hot flashes and vaginal dryness and preventing osteoporosis. However, large clinical trials revealed that certain HRT regimens could increase the risk of other conditions, including blood clots, strokes, and certain types of cancer, like breast cancer and uterine cancer if estrogen is given alone. So the decision to use HRT is now much more individualized, weighing the potential benefits against the risks for each woman.
Okay, let's shift now to what happens when reproduction is successful. Fertilization and the beginning of pregnancy. There's a fertile window, right? Sperm and egg don't last forever.
Correct. Sperm can survive in the female reproductive tract for up to about 4 to 6 days, potentially. The egg, after ovulation, is only viable for fertilization for about 24 to 48 hours. So for fertilization to occur, intercourse needs to happen sometime between roughly 5 days before ovulation and 1-2 days after ovulation. That's the fertile window.
Where does the egg go after ovulation?
The fimbriae at the end of the fallopian tube sweep over the ovary and help capture the ovulated secondary oocyte. Cilia, lining the inside of the fallopian tube, then slowly propel the egg towards the uterus. This journey takes about 4 days. Fertilization usually occurs within the fallopian tube, typically in the outer third, the ampulla.
And the sperm's journey. Millions are ejaculated, but how many reach the egg?
It's an arduous journey for sperm. Of the hundreds of millions deposited in the vagina, they have to navigate the cervical mucus, which is thin and watery around ovulation due to estrogen, travel up through the uterus, and into the correct fallopian tube. There's massive attrition along the way. Only a few hundred, maybe just 100, 200, actually reach the vicinity of the egg. And during their transit through the female tract, sperm undergo a process called capacitation. This involves changes to their motility pattern, they become hyperactive, and alterations to their plasma membrane, making them capable of fertilizing the egg. They can't fertilize immediately after ejaculation.
Okay, sperm meets egg in the fallopian tube. What happens right at fertilization?
Many capacitated sperm may reach the egg and bind to receptors on its outer coating, the zona pellucida. This binding triggers the acrosome reaction in the sperm head. Remember the acrosome. It releases powerful enzymes that digest a path through the zona pellucida. It's like drilling through the shell. Usually, the first sperm to successfully penetrate the zona pellucida and reach the egg's plasma membrane fuses with it. The sperm nucleus and centriole then enters the egg cytoplasm.
Only one sperm should get in, right? What stops others?
Absolutely critical to prevent polyspermy fertilization by more than one sperm, which leads to a non-viable embryo. There are two main blocks. First, a fast block. The fusion of the first sperm instantly changes the electrical potential across the egg's membrane, making it unreceptive to other sperm for a short time. Second, a slow block, which is more permanent. Sperm fusion triggers the cortical reaction. Vesicles just beneath the egg membrane, cortical granules, release their contents outwards. These enzymes alter the zona pellucida receptors so no more sperm can bind, and they harden the zona pellucida, making it impenetrable.
Clever defense system. So the sperm nucleus is inside. What happens then?
The entry of the sperm triggers the secondary oocyte to complete meiosis II, forming the mature ovum nucleus, the female pronucleus, and the second polar body. The sperm nucleus decondenses to form the male pronucleus. Both pronuclei, each containing 23 chromosomes, replicate their DNA. Then, the two pronuclei migrate towards each other, their membranes break down, and the maternal and paternal chromosomes mingle on a single spindle. This fusion marks the completion of fertilization, creating the single-celled zygote with 46 chromosomes, and it triggers the start of embryonic development, or embryogenesis.
And this zygote is still in the fallopian tube.
Yes, it starts dividing as it continues its journey towards the uterus.
What if it implants somewhere else, like in the tube itself?
That's an ectopic pregnancy. Most commonly, implantation occurs in the fallopian tube. This is a dangerous situation because the tube cannot accommodate a growing pregnancy and can rupture, causing severe internal bleeding. Ectopic pregnancies are not viable and require medical intervention.
Okay, assuming it reaches the uterus safely. What happens between fertilization and implantation?
As the zygote travels down the fallopian tube, it undergoes a series of rapid mitotic cell divisions called cleavage. The cells divide, but the overall size of the conceptus, the collective term for everything derived from the zygote, doesn't increase much initially. By the time it reaches the uterus, around 3 to 4 days after fertilization, it's typically a solid ball of 1632 cells called morula. These early cells are considered totipottent, meaning each one could potentially develop into a complete individual to a city. This is how identical twins can form if they split early. Over the next couple of days floating in the uterus, the morula develops into a blastocyst. This stage, around 100 cells, is characterized by differentiation. There's an outer layer of cells called the trophoblast, which will contribute to the placenta, and an inner cluster of cells called the inner cell mass, which will form the embryo, proper, and fetus. There's also a fluid-filled cavity.
So blastocyst is ready to implant. When does that happen?
Implantation typically begins around day 20 or 21 of the menstrual cycle, which is about 6 or 7 days after ovulation and fertilization. The uterus, under the influence of progesterone from the corpus luteum, secretory phase, is receptive at this time. The blastocyst adheres to the endometrial lining, and the trophoblast cells start to proliferate rapidly. They secrete enzymes that digest the endometrial tissue, allowing the blastocyst to burrow into and embed within the uterine wall.
This leads to the formation of the placenta.
Yes, placentation begins during implantation. The placenta is a remarkable organ formed from both fetal tissue, derived from the trophoblast, which forms structures called the chorion and chorionic villi, and maternal tissue, the modified endometrium, now called the decidua. These fetal and maternal tissues become intricately interlocked. The chorionic villi contain fetal capillaries, and they project into maternal blood-filled spaces within the decidua.
Does the blood actually mix?
No, critically, fetal and maternal blood do not mix directly. Exchange occurs across the thin barrier formed by the cells of the chorionic villi and the fetal capillary walls. Nutrients, oxygen, antibodies pass from mother to fetus. Waste products like carbon dioxide and urea pass from fetus to mother. Hormones are also exchanged and produced by the placenta itself. The umbilical cord develops, connecting the fetus to the placenta, containing the umbilical arteries and vein that carry fetal blood back and forth. Meanwhile, a membrane called the amnion develops around the embryo, enclosing the amniotic cavity, which fills with amniotic fluid. This fluid cushions the fetus, allows movement, and maintains temperature.
This intricate connection, the maternal-fetal unit, is obviously vital and vulnerable.
Very vulnerable. This is why maternal nutrition is so critical during pregnancy. Things like adequate folate intake are essential to prevent neural tube defects in the developing embryo. It's also when the fetus is susceptible to teratogens agents like certain drugs, for example, alcohol, thalidomide, chemicals, or viruses, for example, rubella, that can cross the placenta and cause birth defects.
And that idea of epigenetic programming applies here too.
Absolutely. The maternal environment, nutrition, stress, exposure to toxins can influence fetal gene expression patterns with potential long-term consequences for the offspring's health, as we discussed. And the integrity of the placental barrier is also crucial for protecting the fetus, which is genetically different from the mother, from being rejected by the mother's immune system.
This leads to methods for checking on the fetus, right? Fetal diagnosis.
Yes. Techniques like ultrasound provide images. Amniocentesis involves sampling amniotic fluid to test fetal cells for genetic abnormalities. Chorionic villus sampling, CVS, samples placental tissue earlier in pregnancy but carries a slightly higher risk. And increasingly, maternal blood screening can detect fetal DNA fragments or specific markers, like proteins associated with Down syndrome, circulating in the mother's blood, offering non-invasive screening options. #SEX17.21 hormonal and other changes during pregnancy.
Finally, pregnancy itself requires major hormonal adjustments to maintain it, right?
Huge adjustments. The most striking change is the continuous dramatic increase in plasma levels of both estrogen and progesterone throughout the 9-month of gestation.
Where do these hormones come from? Still the corpus luteum?
Initially, yes. For the first 2 months or so, the corpus luteum, rescued from degeneration by a signal from the implanting embryo, is the primary source. But then the placenta gradually takes over as the main producer of both estrogen and progesterone for the remainder of the pregnancy. The placenta becomes a major endocrine organ.
What are these high levels of estrogen and progesterone doing?
Estrogen promotes the continued growth of the uterine muscle, myometrium, needed to accommodate the growing fetus and for eventual labor contractions. It also stimulates breast duct development. Progesterone is absolutely essential for maintaining the pregnancy. It suppresses contractions of the uterine smooth muscle, preventing premature expulsion of the fetus. It maintains the uterine lining, decidua. It also helps suppress the maternal immune response against the fetus. It's often called the hormone of pregnancy.
What about the pituitary hormones, FSH and LH?
During pregnancy, secretion of FSH and LH from the mother's anterior pituitary is very strongly inhibited by the high levels of estrogen and progesterone, and possibly placental inhibin. This prevents any new ovarian follicular development or ovulation during pregnancy.
But the pituitary does increase secretion of another hormone.
Yes. Prolactin secretion from the anterior pituitary increases progressively throughout pregnancy, stimulated by the high estrogen levels. Prolactin prepares the breasts for milk production, although actual milk secretion is usually held in check by the high estrogen and progesterone until after delivery. #tag outro.
And that wraps up our deep dive into the truly fascinating world of human reproduction, drawing from Chapter 17 of Vander's Human Physiology. We've covered, well, quite a lot, from the basics of gamete formation and sex determination, all the way through the intricate hormonal cycles, the amazing event of fertilization, and that incredible journey of early development in pregnancy.
Yeah, it's a complex area. We really hope this exploration has helped clarify some of these vital physiological processes for you, hopefully providing some of those aha moments and the specific details you might need to feel, genuinely well-informed on the topic.
We certainly learned a lot revisiting it. Thank you so much for joining us on this deep dive.
And from the entire team here, we wish you continued curiosity and lots of discovery in your studies.
Goodbye for now.